Today we're talking about Chapter 7, Section 4, which is about pedigrees. A pedigree is simply a diagram that shows the family history of a certain trait. It could be a disease like hemophilia that's shown here, or maybe you're just showing who in the family has blue eyes. Whatever your trait is that you're interested in, you can make a pedigree for it. These are the symbols that you would use on a pedigree. A square is going to represent a male, and a circle represents a female. If we shade that square or circle in, that would indicate the person has the trait that you're interested in. So we would call that an affected male or affected female. If you see a diamond, that simply means that the person, their sex is not known. Maybe it's an unborn child. Maybe it's a group of 2 or 3 siblings and it's not specifying whether they're male or female. That's a pretty rare symbol to see on a pedigree, but you might run across it. A horizontal line represents a marriage or mating between a male and a female. That is very common on a pedigree. That shows who has offspring with whom. And if you see a double horizontal line, that's what we call a consanguineous marriage, and that would be a marriage between closely related individuals. Maybe they're cousins or maybe even siblings. This is more common in some of the historical pedigrees, maybe in a royal bloodline or something like that, where they're trying to not mix blood, so to speak, with other families. A vertical line is going to represent offspring. So from that marriage line here, we have the offspring line. So this would be the son of these 2 parents. If there's multiple children, they would all come down from this one vertical line, but then obviously we need to spread them out so we can see their shapes. These 2 representations would show twins, which again are pretty rare on a pedigree. And then finally, this last symbol, this is only shown on some pedigrees, and the half-shaded shapes would represent someone who is a carrier. Again, not every pedigree is going to use this, so if you don't see half-shaded shapes, it doesn't necessarily mean that no one in the pedigree is a carrier. It could very well mean that it's impossible to know if you're a carrier or not. And so if you're making a pedigree, maybe you can only shade in the shapes that you know have the trait, but you don't know for sure if someone's a carrier, so you would leave the shading off. Now you read a pedigree by, first of all, looking at the generation. You can see that they are numbered on the left side using Roman numerals from 1 through 4. And then the individuals in each generation are numbered under their shapes. So if we want to talk about this individual right here, we could very easily refer to him as generation 3-2. So we would use 3-2 to mean that individual. So here's a chance to practice. I'll put some questions up. If you need to pause the video momentarily to think of your answer, then please do that, and then you can hit play to see if you are right. First of all, how many males are present in this pedigree? Well, you figure that out by counting the squares, and you should get 13. Same for the number of females, except now, obviously, the shape for the female is a circle. You should have found 16. We're not including any of the diamond shapes because those aren't specified male or female, so we can't include them in our totals. Now, how many females actually show the trait or are affected by the trait being studied? Hopefully you got 6. There were 6 circles that were completely shaded in, meaning those were the females that were affected. Next, what is the sex of offspring 3-9? Well, if you look, 3-9 is right here. That is going to be male. That is a male offspring. And finally, how many offspring did the generation 1 parents have? Correct answer is 5. If you said 8, you were looking at every individual in this generation, but remember, only the offspring are going to be connected by the vertical line. So we have 1 here, 2, 3, 4, 5. This person 2-8, for example, is the wife of person 2-7 and not technically the daughter of those 2 original parents. So you have to pay attention to who is connected with those vertical lines. Now let's talk about how to interpret a pedigree in terms of genotypes. You can use a phenotype, the physical expression of a trait, to infer genotypes on a pedigree by looking at the patterns between parents and their offspring. You are going to notice, and we'll talk about several examples here, that an autosomal gene or a trait controlled by a gene located on chromosomes 1 through 22, those are going to show different patterns than a sex-linked gene. Remember, sex-linked traits are found on a sex chromosome, either X or Y, most of the time X. In this pedigree, we are told that the person number 5 in the F1 generation is little w, little w. They are homozygous recessive, and their shape is unshaded. So that means all of these shaded in shapes with the blue coloring, they must have a dominant phenotype. And that means that any shaded shape in this pedigree has to have at least one dominant capital W allele. So we are already certain that this pedigree is showing the inheritance of a dominant trait. Now, if we use this information to infer genotypes and we start with person 1, the father in the parental generation, can we tell his genotype? And the answer is yes, we can, because he is unshaded just like person 5 is unshaded, so he has to be homozygous recessive just like person number 5. Now let's look at person 2, the mother. She has a blue shaded circle, so we know she has at least one dominant allele. Can we tell if she has a second dominant allele? Well, the answer is yes if you look at the offspring. Again, individual 5 is homozygous recessive. This person has inherited 2 recessive alleles. Obviously, one came from dad and the other one had to come from mom. So mom's genotype must be heterozygous for this trait. So go ahead and see if you can predict the genotypes for the rest of the Punnett square. Go ahead and pause the video while you do that. And then once you hit play, you can see if you got the correct answers. All right, now we're going to look at individual number 3. Individual number 3 was an easy one, homozygous recessive. Again, unshaded, therefore they're showing the recessive phenotype, so they must have 2 lowercase w alleles. Now on to individual number 4. This person has to be heterozygous, and the way we know that this person is heterozygous is we look at her parents. Dad was homozygous recessive. That means dad had to pass down a recessive allele to all of his offspring. There's no way that person number 4 could have 2 dominant alleles because dad only had recessive alleles to pass down. So that's how we know person 4 is heterozygous. We use the same reasoning for person 6. Person 6 also has to be heterozygous because he has the same dad as person number 4. Now, if we look at person number 7, we know that she has a dominant phenotype. So she has at least one capital W, but we can't tell what her second allele is. We just don't have enough information because we can't see her parents, and all of her offspring are showing a dominant phenotype. So all we know is she has at least one capital W, but we don't know her second allele. Now on to the F2 generation. Person 8 has to be heterozygous, for the same reason why person 4 had to be heterozygous. If you look at person 8 dad, that is number 3, and he was homozygous recessive, so he had to pass down a recessive allele to all of his offspring. Person 9 must be homozygous recessive because this is an unshaded shape, so the only way you could have that is if you are homozygous recessive. Person 10 must be heterozygous for the same reason as person 8. The person 10 got a recessive allele from dad. Now let's look at 11, 12, and 13. All we know is they have a dominant phenotype. What we don't know is what their second allele is. We just don't have enough information based on the pedigree given to us. So they could be either homozygous dominant or heterozygous. Now we're going to go on to some examples. Our first example pedigree is autosomal dominant. So here's some information to consider about pedigree patterns that you're looking for. If the trait is autosomal dominant, males and females are going to be equally affected, and you are going to be looking for a trait to most likely show up in every generation because a dominant trait, you only need one allele to express it. So it is very common for a dominant trait to show up in every single generation. In order for offspring to have the trait, at least one parent must have it. So let's look at person 5 here. Person 5, his mom does not have the trait. If this is dominant, that means he had to get a dominant allele from someone, and that someone must be dad. So dad was affected. Same here. This person 3 was affected. They had to get a dominant allele from one of their parents. And so that's why you see it showing up in every generation. An example of this is polydactyly, which is having extra digits. That's actually a dominant trait, as well as Huntington's disease. Both of these are caused by a dominant allele, but both of them are fairly rare in the population. So don't get into the habit of thinking that if something is rare, it must be recessive. It can absolutely still be a dominant trait. Our next example would be autosomal recessive. At first glance, you can already see how these pedigrees look different. Again, if it's autosomal, we have equal likelihood of males and females being affected. But if it's recessive, it can absolutely skip a generation. And the typical thing that you're looking for here is if you have an offspring that has a trait coming from parents who don't. Whenever you see affected offspring coming from 2 unaffected parents, you have to be looking at a recessive trait. The only way that this could happen would be if both parents pass down a recessive allele, then their offspring could be affected even though neither parent was. One important thing to note, just because one offspring is affected, that does not impact the chances of other offspring inheriting the disorder. You always make that prediction from a Punnett square. So if there's a 1/4 chance of one child having it, if that couple were to have a second child, it's still a 1/4 chance. It doesn't reduce the risk in the second child just because the first child ended up having it. And some examples here of autosomal recessive conditions would be cystic fibrosis, which is a lung disease primarily. You get a lot of thick, sticky mucus built up in your lungs. And also Tay-Sachs disease, which is a lysosome disease. The lysosomes are unable to break down certain fats in the brain, and that is normally fatal by the time a child would be the age of 5. Our next pattern is X-linked recessive. This is going to be an example of a sex-linked trait because the allele is located on a sex chromosome. Particularly in this case, it's on the X because we're calling it X-linked. That is definitely the most common sex chromosome to have an allele on it for a particular disorder. Now, because it's sex-linked, we do see males being more affected than females, and because it's recessive, we can see it skip a generation and show up in offspring whose parents did not have that particular trait. So that's what's happening here. Individual 3-2 has inherited one recessive allele from the mother, and that is why he has the disorder, but neither parent did. Mom happened to be a carrier in this case. You're never going to see an X-linked recessive disease be passed from father to son, and that is simply a matter of the fact that fathers do not pass X chromosomes to their sons. They will only pass their Y chromosome, and that's what makes the baby a son, makes him a boy. So in this case, you are not going to see a father passing down a trait to his sons. Affected sons have inherited that from their mother. So your typical pattern that you see for X-linked recessive pedigree is when the grandfather has a condition, he is going to produce daughters who are carriers because all of his daughters inherit his X, which is having an allele for the disorder. And then those daughters have a 50% chance of passing down that X to their sons. So about half of the grandsons of that grandfather would also be affected. And that's what you see in this pedigree here. Examples of X-linked recessive disorders would be things like color blindness. If you cannot distinguish between red and green, that's the most common type of color blindness. And also Duchenne muscular dystrophy, which is a muscle deterioration disorder. Unfortunately, there's not really a treatment or a cure for that. So your muscles, once they deteriorate, you never are able to regain that function. Next, we have X-linked dominant. An X-linked dominant pedigree is not something that's very common. There are not a whole lot of X-linked dominant disorders. But it is still sex-linked because the disorder has an allele on the X chromosome. But in this one, females are more likely to be affected than males. And that's because females get 2 X chromosomes. So they have 2 opportunities of inheriting a bad chromosome instead of the males who only have 1 opportunity to inherit a bad X. Now this one's dominant, so we do see a typical pattern of it tending to show up in every generation or most generations. It actually doesn't show up in this very final generation at the bottom. That's okay. It just so happens that those parents weren't affected at all, so they produce offspring that were unaffected. But it is showing up in each of those other 4 generations. So generally speaking, it tends to show up in each generation. Now, in this case, a male is going to pass it down to all of his female children because, again, the male always passes his X down to his daughters. If this is a dominant allele on the X chromosome, all of his daughters will inherit 1 copy of a dominant allele and will be affected. And those affected females who get 1 allele from dad are going to have a 50% chance of passing it down to their offspring. And so that's why it's very common to show up in every single generation. Now, generally speaking, this is a rare pattern. You're not going to see a lot of Punnett squares, pedigrees that show an X-linked dominant pattern. And there aren't a whole lot of diseases that are caused by this. One example is called double cortex syndrome, and it has to do with brain development. You can see that this brain on the right, it looks like it has a double layer around it. So that kind of crowds the brain as the brain is developing and can have severe consequences in terms of intelligence and learning. All right, next we have a Y-linked pedigree. In a Y-linked pedigree, this is even more rare than X-linked dominant. So in a Y-linked pedigree, you can literally only have males affected because it is a Y chromosome that's impacted and females don't ever inherit a Y chromosome. So we don't really even have to designate it as dominant or recessive because a male only has 1 Y. If he has the allele for the trait, he's going to express the trait. This disease is always going to be passed from father to son because a father always will pass his Y chromosome to his sons. That is what makes them develop as male. This is extremely rare. There's only around 60 genes on the Y chromosome to begin with versus the 800 you would have on the X. And so it is unlikely that those 60 genes would have something happen to them to cause a Y-linked disorder. One example of Y-linked inheritance is an interesting one, hypertrichosis pinnae, or hairy ears. It has shown to be caused by a gene on the Y chromosome and therefore be Y-linked. And then the other example is not really a disease, it's just an example of genes on the Y chromosome that are passed down from fathers to sons. And this is a group of genes called SRY. And that SRY group of genes is essentially the genes that make a male develop as male. So obviously that set of genes always gets passed down from father to son. so that the sons of a father are going to develop male sex characteristics. Our final example here is non-nuclear inheritance. And this one is actually really easy to pick out on a pedigree if you know what to look for. Remember that non-nuclear means the trait is not in the nucleus, so it's in the genes of the chloroplast or the mitochondria. So the mother is the source of all of those organelles. It's the egg cell of the mother that contains the cytoplasm and the organelles that go on to become all of the organelles in the offspring. So in this case, we have 100% inheritance from the mother. We don't really have to call it dominant or recessive because basically you either have the trait because of your mother's mitochondria or you don't have the trait. All of the offspring from a female parent will be affected by this trait. So there is a mitochondrial disease in humans that is only passed down from mother to offspring, and all of her offspring would have it. You also have the leaf pigment gene in chloroplasts of plants that when that is passed down from the mother, all of the offspring have it. So you can see that pattern here. This affected mother has 3 affected offspring. Her son does not pass it down at all, but her daughter does. Every single one of those offspring are affected because they all got their mitochondria from the mother. So that's our final example of pedigree patterns that you need to look out for. This is going to become much more easy the more practice you do, so you'll have ample opportunity to practice in class. But as always, if you are struggling and you have questions about this content, please make sure you ask about that in class and we will get it sorted out.